Hi everyone, welcome to fourth series of bacteriology. In this lecture, we are going to see about many things like capsule, cell wall, cytoplasmic membrane, then protoplasm, nuclear material, then inclusion granule, then flagella, Cambrian, porphyry, bacterial spore. Then we will finally will discuss about chemical composition of bacteria. One by one, we will take each and every topic. So like about capsule. Capsule, I will start from outside. The capsule is outermost layer, then cell wall, like that. In that scenario, we will cover each and everything. So without any delay, let's get started. So दिस इज सिंपल स्ट्रक्चर ऑफ बैक्टीरिया। आप इस ब्लैक बोर्ड पे देख सकते हैं, ये एक सिंपल स्ट्रक्चर ऑफ बैक्टीरिया है। हम यहाँ पे गौर से देखेंगे तो हमें एक बात समझ में आएंगी। वो है यहाँ पे। सो ये जो आउटर लेयर है, ये सबसे आउटर मोस्ट लेयर, हम उसको क्या बोलते हैं? कैप्सूल। ये कुछ बैक्टीरिया में ये कैप्सूल लेयर होता है, कुछ बैक्टीरिया में नहीं होता है। सो हम देखते हैं स्टार्टिंग का सबसे आउटर मोस्ट लेयर इस क्या होता है, इसको क्या बोलते हैं? कैप्सूल। सो कैप्सूल को हम डिटेल में समझेंगे क्या केमिकल कंपोजिशन है कैप्सूल का। 2% सॉलिड है, ये 2% सॉलिड है, और हम बोल सकते हैं 98% वाटरी बोल सकते हैं हम इसको। जिलेटिनियस कवरिंग है। ये जो कवरिंग है होती है बैक्टीरिया की, तो आउटर मोस्ट कवरिंग ऑफ दिस बैक्टीरिया इज अ gelatinous covering and capsular gel which is filled with the capsular gel. In that chemical composition of this capsule is about complex of polysaccharides, polypeptide and protein. These three molecules mainly are present in this capsule. So and the capsular gel, it is filled with the capsular gel. And in some bacteria there is a capsule. कुछ बैक्टीरिया में कैप्सूल होता है, हम उसको क्या बोलते हैं? कैप्सुलर बैक्टीरिया। और कुछ बैक्टीरिया में कैप्सूल नहीं होता है, तो हम उसे क्या बोलते हैं? अनकैप्सुलेटेड बैक्टीरिया। 2% सॉलिड मीन्स इसमें 98% जेन्युइनली लिक्विड होगा ही। सो हम इसको बोलते हैं जिलेटिनियस कवरिंग। इसका बहुत अच्छा से वर्क है। ये जिलेटिनस कवरिंग उसको प्रोटेक्टेड करता है, एक सी काइंड ऑफ कैप्सूल कोट प्रोवाइड करता है। और इसके वजह से जो बैक्टीरिया का जो बोल सकते हैं डिकम्पोजिशन या बर्स्टिंग होना पॉसिबल नहीं है एज यूजुअल। मेनी फंक्शन करते हैं, जैसे कि फूड के जब बैक्टीरिया को छुपना होता है, लाइक बैक्टीरिया व्हेन सम वांट टू हाइड themselves, this kind of capsular shell somewhat help in that process. Then this is the what is the ropiness? At the end this point is remaining ropiness. The growth of capsule forming bacteria cause the type of spoilage इज कॉल्ड एज रोपिनिस। जब बैक्टीरिया का ग्रोथ होता है एक तरह से, तो जब वो स्पॉइलेज करता है उसे हम क्या बोलते हैं? रोपिनेस बोलते हैं। व्हेन द ग्रोथ ऑफ कैप्सूल फॉर्मिंग बैक्टीरिया कॉजेज द स्पॉइलेज। स्पेसिफिकली जब जो कैप्सुलेटेड बैक्टीरिया होता है, जिसके ऊपर कैप्सूल होता है, स्पेसिफिकली वो बैक्टीरिया जब एक काइंड ऑफ स्पॉइलेज कॉज करते हैं इन फिश के टर्म्स में। जब एक आधी एग्जांपल ले सकते हैं, हमने फिश ले आया मार्केट से और वो फिश को हमने क्या किया? कहीं पे तो रख दिया। जब ये बैक्टीरिया कैप्सुलेटेड बैक्टीरिया जो बोलते हैं हम इसको कैप्सूल फॉर्मिंग बैक्टीरिया, व्हेन वी आर कॉलिंग अबाउट दिस बैक्टीरिया स्पेसिफिकली अबाउट कैप्सुलेटेड बैक्टीरिया, व्हेन दे कॉज spoilage, that's the particularly of that spoilage is we called it as ropiness. जब particularly कैप्सुलेटेड ही बैक्टीरिया जब ये करता है, स्पॉइलेज करता है, हम उसको क्या बोलते हैं? रोपिनेस ये बोलते हैं। सो नेक्स्ट हम देखते हैं यहाँ पे, दिस इज ओवरऑल अबाउट कैप्सूल। नेक्स्ट हम देखते हैं यहाँ पे द ग्रोथ ऑफ कैप्सूल फॉर्मिंग बैक्टीरिया कॉज रोपिनेस, हमने ये डिस्कस किया है। सो नेक्स्ट वी विल सी अबाउट सेल वॉल। सो सेल वॉल क्या होता है? हमने पिछले लेक्चर में इन बैक्टीरियोलॉजी 03 लेक्चर में हमने देखा था कि जो कवरिंग होती है सेल वॉल की क्या कैसे होती है, सेल वॉल कैसे मेडअप होती है। ग्राम स्टेनिंग हमने वहां पे सीखा, ग्राम पॉजिटिव ग्राम नेगेटिव बैक्टीरिया के बारे में जाना, फंक्शन ऑफ द सेल वॉल जाना। बट वी आर कंप्लीटिंग वन मोर टाइम इन शॉर्ट पॉइंट। ओके, सो लेट्स स्टार्ट। A cell wall 10 to 25 micrometer is thick. So by point of JRF, this is most important. MCQ आ सकता है एग्जाम में कि 10 to 25 micrometer centimeter, what is the diameter of the cell wall, particularly bacterial cell wall? So option will given like that, 4 to 5 micrometer in diameter, 6 to 7 micrometer in diameter, like randomly, and 10 to 25 micrometer in diameter. So we have to point or mark that specifically 10 to 25 micrometer in thick. This cell wall is elasticity means any material which is elastic, we call it as elasticity property, support plasma membrane. Already in 03 lecture I explained about how they cell wall support the plasma membrane, and about from internal osmotic pressure, how the internal osmotic pressure works in the cell wall. It consists of measure cup macromolecule, म्यूकोप्रोटीन पॉलीसैकेराइड चेन एंड पेप्टाइड ब्रिज. सो मैंने पहले ही लेक्चर में आपको बताया था 03 लेक्चर में कि कैसे मैक्रोमोलिक्यूल म्यूकोप्रोटीन पॉलीसैकेराइड कैसे अरेंज होते हैं एक दूसरे के साथ, कॉम्पैक्ट कैसे प्रोवाइड करते हैं, और इस कॉम्पैक्ट या अरेंजमेंट की वजह से रिजिडिटी आती है सेल वॉल को. सो नेक्स्ट वी विल डिस्कस अबाउट नेक्स्ट इनर. We are going like that from outer to inner, we are entering in the cell. So let's cover one by one. So this is a cytoplasmic membrane. The cursor, like you can say, यहाँ पे जो देख रहे हैं आप, यहाँ मेरा कर्सर मूव कर रहा है. सो यहाँ पे जो दिख रहा है ये जो कलर, हम स्पेसिफिकली साइटोप्लाज्मिक मेम्ब्रेन है. ये साइटोप्लाज्मिक मेम्ब्रेन कैसे होता है? साइटोप्लाज्मिक मेम्ब्रेन इट कवर प्रोटोप्लाज्म. ये जो साइटोप्लाज्मिक मेम्ब्रेन बोल सकते हैं, हमने पहले क्या देखा, आउटर से इनर तक. आउटर साइड में क्या था? कैप्सूल था. फिर हमने क्या देखा? सेल वॉल देखा. फिर हमने क्या देखा? अब हम देख रहे हैं साइटोप्लाज्मिक मेम्ब्रेन. साइटोप्लाज्मिक मेम्ब्रेन इट कवर्स प्रोटोप्लाज्म. द ऐसे क्वेश्चन आ सकता है इन एग्जाम की, विच इज द पार्ट विच कवर्स द साइटोप्लाज्म, विच प्रोटोप्लाज्म, दैट पार्ट इज साइटोप्लाज्मिक मेम्ब्रेन. It covers protoplasm. It is about 0.5 to, it is about 0.5, it is about 0.5 to 10 macron in thick, and it consists of lipoprotein. Lipoprotein means lipid and protein. You can see here this outer structure is lipid, like a lipoprotein, lipid, and the inner is protein like structure, lipoprotein structure like structure. Its main function is active selection and transport of nutrient solute. Active selection means something which is required for the cell. This channel, you can say this channel, passing channel, yeah, protein gated channel. This, through this protein gated channel, something which is present outside, if they require for bacteria, it is selectively get pushed or get allowed by the cell inside this cell. So transport of nutrient solute, its main function is active selection and transport of nutrient solute. The membrane itself has little mechanical strength and it's supported by the external cell wall. ये मेम्ब्रेन को हम बता सकते हैं, मेम्ब्रेन को जो सपोर्ट करता है वो एक्सटर्नल सेल वॉल होता है. वी हैड ऑलरेडी डिस्कस दिस. This is overall about cytoplasmic membrane. Next we will see about next organelle. So here is protoplasm. Now we will discuss about complex structure, not complex bit, but it is interesting structure, or protoplasm. It is interesting structure of the cell wall. It is interesting structure of the protoplasm. In the protoplasm, viscous watery solution or soft gel is present, contain organic and inorganic solute. In this protoplasm, there is a organic and inorganic solute are present. This kind of black structure is called organic and inorganic, like that. Just memory map created by me. So you can see organic and inorganic both molecules are present in protoplasm. It filled with the tiny granule 10 to 30 micrometer diameter called as ribosome. So you can say 10 to 30 micrometer diameter called ribosomes are present in that. These are the ribosomes, which is 10 to 30 micrometer diameter called ribosomes are present in this protoplasm. RNA is the site of protein synthesis in the cell. This is RNA. This RNA ribonucleic acid. This RNA is the site of protein synthesis in the cell. This is specifically about protoplasm. Many things occurs in protoplasm, specifically in eukaryotic and prokaryotics. We are talking about here prokaryotics. In case of eukaryotics, there is particularly you can say the DNA is present in that nucleus. So this is about overall about protoplasm. Next we will discuss about next part of the cell, nuclear material. It is a deoxyribonucleic acid. Nuclear material is a DNA in specifically in this protoplasm. Now we will discuss about inclusion granule. What are these inclusion granule? Inclusion granules are round granule sorry, inclusion granule round granule found in the protoplasm. These are the inclusion granules which are round in structure like this, which are found in protoplasm. Not essential structure, which is not much essential structure. Some excess metabolite is stored as a reserve nutrient. It consists of lipid. And this inclusion granules, stored nutrient are reserved material, like which is not that useful for the cell, which are stored in the excessive material, which are stored in the form of lipid, glycogen, polysaccharide, and sulfur in this inclusion granule. This is about inclusion granule. We are seeing each and every like particular part one by one. So next part is that next part is flagella. In that flagella, you can see this is a flagella, but there is a mistake here. This flagella should be marked here. Flagella. This is flagella. 0.2 macrometer macron thick, long as a bacterial cell, originated from bacterial cell wall. It is originated from bacterial cell wall, particularly from protoplasm. 9 plus 2, 9 plus 0 arrangement is flagella are present in different bacteria and different. So peritrichous, eukaryotics, and prokaryotes flagella arrangement is different. So it's not no need to understand here in deep, because in biochemistry we will discuss all these things again about cell and cell wall function like that. Peritrichous flagella arrange over all the body. When flagella present all over the body, such bacteria we called as peritrichous. And again, again on the flagellar movement and flagellar arrangement, there are different types of bacteria, that is peritrichous, lophotrichous. Then at some bacteria there is a flagella is present at the one pole, so it is different kind of names on that particularly. At the end or both end, some bacteria is present at one end or both end. Here also present, here also present. Mm, Polar flagella may aggregate into bundle and forms a tail. When sometimes this in bacteria, the polar flagella, these are the polar flagella, like this flagella form at form. These flagella form like this structure, and then they form and form a tail-like structure in some bacteria. Flagella consist or largely or entirely of a protein flagellin similar to myosin. This is important. Flagella consist largely entirely of protein. Flagella mostly contain or entirely contain flagellin protein, which is like a myosin, actin and myosin. In past lecture, we had discussed that about flagellin protein, how the flagellin protein and all, how there's arrangement like that. So this is all about flagella. There is nothing important in this point. The most important thing is what is the thickness of flagella by GRA point of view, and peritrichous and flagellin protein, how flagella is made up. That is about flagellin protein. So next we will discuss about fimri or pili. Let me clear this. So fimri or so here is a fimri. Here is fimri or pili or gram negative. Bacteria possess very fine hair-like appendage called as fimri. In some gram negative bacteria, there is a fine hair-like appendage called as fimri. You can see this is a fimri. These are the fimri-like structure. Fine hair-like. This is most important structure. What is important here? In exam, the question will be like this: which part of the cell wall is a fine hair-like structure? That is a Pimbri or Pili will be in the MCQ. You have to mark it down. So it is about Fimbri or Pili. They are peritrichously. They are peritrichous means it present all over the body. Single cell have up to 500 Fimbri. That's important, because in single cell, see this important. How many Fimbri or Pili present in single bacteria all over the body? It is about 500 fimri present all over the body in randomly structured. It is not well mannered or like particularly, it is randomly present all over the body. They have adhesive property. See, this is a small bacteria, and this bacteria need to be attached with the rock or substratum or with the food, you can say. Let me change my color. So this is a, you can say this is a rock. Yeah, rock, or you can say food or something which is material, you can say. This bacteria have this kind of fimbri or pili, this kind of fimbri or pili. Due to this fimbri or pili, they get attached with them, they get attached with the substratum or with the rock, and they consume that food due to air resistance or due to some kind of, we can say, it get not removed by that. So it get attached and consume that food due to fimbriae or pili. They get kind of adhesive property in fimbriae. That is the most important. So next we will discuss about bacterial spore. Bacterial spore. About bacterial spore, if I talk about bacterial spore, then spore. This is a bacterial spore. In the bacterial spore, you can say this in the bacterial spore, Colastridium and Bacillus develop a resting phase called a endospore. Some specifically bacteria, Clostridium and Bacillus. This bacteria develop a endospore. That endospore form under adverse condition. Spore formed in the protoplasm of the parent vegetative cell. These spores are formed in the parent and vegetative cell. Parent vegetative cell. Example, if I want to tell you something here, let me take my pen. Yeah. So, uh, yeah, there is a scarcity of food. This is a bacteria, and this bacteria requires a food. This bacteria is a require food, but what happens, this food is not here. दैट फूड इज नहीं है यहाँ पे, हम बोल सकते हैं यहाँ पे फूड की अवेलेबिलिटी नहीं है उतनी अच्छी खासी. सो क्या होता है, जब ऐसे फूड की अवेलेबिलिटी नहीं होती है इस समय पे, क्या करते हैं बैक्टीरिया, उसके अंदर एक स्पोर फॉर्म कर लेते हैं. दैट इज एंडोस्पोर. ये किस वजह से फॉर्म कर लेते हैं कि यहाँ पे फूड की कमी है. अगर उन्होंने डिवीजन कर भी लिया, एट दी एंड उनकी जो प्रोजेनी या उनकी जो डॉटर सेल है, एट दी एंड वो सर्वाइव करने वाली नहीं है कैसे भी. सो इन सच सिचुएशन दे फॉर्म अ इंडोस्पोर inside that cell. Like this, they form endospore. That endospore, what the characteristics of that endospore is that keep that cell, that endospore keep their progeny for survive for long period of duration. जब ऑप्टिमम कंडीशन आएगा, जब फूड की अवेलेबिलिटी होगी, वो स्पोर से उनके स्पोर कोट होती है उसके बाहर. वो कैसे होती है हम नेक्स्ट स्लाइड में देखेंगे. सो ये ऐसे फॉर्म करना मीन्स एंडोस्पोर बोलते हैं. ये स्पेसिफिकली हमें किस में दिखता है, क्लॉस्ट्रिडियम और बैसिलस में दिखता है. From under adverse condition, spore formed in the protoplasm of the parent vegetative cell. The spores are particularly formed in the parent and vegetative cell. So next we will discuss about how this spore and how structure of that spore. So spore coat. This the endospore is look like this. In this endospore, the outermost is the spore is the spore coat. It is a thin and tough outer coat, and inside that part spore coat there is cortex is present. A thick covering layer. It is a thick and covering layer. Let me take a pen. Yeah. It is thick covering layer. This is thick covering layer and membrane. This is a membrane which is present inside the cortex. And core. Core is important. It consists of central core of the condensed protoplasm containing nuclear body. In that core, there is a nuclear body center. At the center is present. It is a core. At the center, there is a nuclear body present. So like this, there is arrangement of endospore is like spore for more resistance to chemical and physical influence than our vegetative cells. They are sensitive to heat only above 100 डिग्री सेल्सियस. जब 100 डिग्री सेल्सियस से ज्यादा हीट होगी तभी ये इसको फर्क पड़ेगा. एट दी एंड 60-70 बोल सकते हैं या 80 डिग्री सेल्सियस तक स्पोर कोट को कुछ टेम्परेचर से फर्क नहीं पड़ेगा उतना ज्यादा. कोर्टेक्स एंड स्पोर कोट इज इम्परमिएबल. ये जो कोर्टेक्स होता है और स्पोर कोट ये इम्परमिएबल होता है, बाहर के वाटर मॉलिक्यूल या बाहर के टेम्परेचर को कोर के अंदर आने नहीं देता. इस वजह से जो भी मटेरियल या कोर में प्रेजेंट है, इट विल बी इट शुड बी it should be in original condition, so it does not get affected by the outer environment. High content of calcium and dipiclonic acid, or very low content of water, and consequent a very low metabolic and enzymatic activity. इसमें जो enzymatic activity होती है वो कम होती है. हाई कंटेंट ऑफ कैल्शियम होता है. ऐसे आ सकता है क्वेश्चन, व्हाट इज द कंटेंट ऑफ एंडोस्पोर लाइक कोर. So high content of calcium and dipiclonic acid present होता है, and low content of water and the consequent very low metabolic enzymatic activity होते हैं. Enzymatic activity low ही होगी और metabolism low ही होगा because they have to survive for long duration. Then the spore germinate when external environment become favorable. जब external environment favorable condition में आता है या जहाँ पे food की availability होती है, तो ये जो बैक्टीरिया स्पोर के आउट से निकल कर स्पोर कोट या ये जो स्पोर कोट एंड कॉर्टेक्स फट जाता है और जो अंदर से कोर से बैक्टीरिया बाहर निकल जाता है लाइक दैट, और कंज्यूम करने लगता है एज नॉर्मल कंडीशन स्टार्ट करता है लाइक दैट. दिस इज ओवरऑल अबाउट स्पोर कोड. And now if we discuss about chemical composition of bacteria, in bacteria 90% is water. In the chemical composition of bacteria, 90% water, specifically hydrogen, carbon, oxygen, and nitrogen constitute 99%. 90% water means H2O. If you see this molecule, H2O, in that H2O, H2O, in this H2O molecule, you see H2. So hydrogen and oxygen are definitely present here. So यहाँ पे बोल सकते हैं H2O 90% प्रेजेंट है, तो H2O में से हम हाइड्रोजन, ऑक्सीजन बोल सकते हैं, और कार्बन और नाइट्रोजन 99% है यहाँ पे. So 10% you can say in what we say. And if you go by chemistry point of view, यहाँ पे 8 to 9% या 9% बोल सकते हैं न कार्बन, देन कार्बन और नाइट्रोजन प्रेजेंट होगा, और 1% दूसरे मेटल प्रेजेंट होंगे, रेयर बोल सकते हैं हम उसको. 99% तो ये हो गया, और 1% सल्फर, फॉस्फोरस, मैग्नीशियम, कॉपर, जिंक, मोलिब्डेनम ये प्रेजेंट होंगे. मोस्ट ऑफ द कार्बन एंड नाइट्रोजन ऑफ द बैक्टीरिया सेल मास आर कंटेन इन द अमाइनो एसिड linked in protein which constitute up to 50% of dry weight of the cell. So question will be like this in JRF: 50% of dry weight of the cell. How the composition of the 50% of the dry weight cell, which is about nitrogen bacterial cell mass are contained in the amino acid. Amino acid हमें पता है, जब many amino acid join together to form protein, amino acid is made up of nitrogen amine group and then a carbon group, and this kind of like structure is amino acid. So 50% of dry weight is of the cell. Other macromolecule consist of the bacteria cell in the descending order are, if we see about other macromolecule in descending order other than this, so it will be like nucleic acid will be present at more, then carbohydrate and lipid. Then if we see about other four macromolecule type are usually found in combination with the other like lipoprotein, then फोर मॉलिक्यूल दे आर लिपिड एंड प्रोटीन कॉम्बिनेशन. देखो यहाँ पे बोला है कि ऐसे मॉलिक्यूल, फोर मैक्रो मॉलिक्यूल जो कॉम्बिनेशन में प्रेजेंट होते हैं. यहाँ पे लिपोप्रोटीन कॉम्बिनेशन में, लिपोपॉलीसैकेराइड कॉम्बिनेशन में, ग्लाइकोप्रोटीन कॉम्बिनेशन में, न्यूक्लियर प्रोटीन कॉम्बिनेशन में, लाइक दिस फोर macromolecular combination. Which are the four macromolecule in combination? This can be question in exam. That will be lipopolysaccharide, glycoprotein, lipoprotein, and nuclear protein. The outer constituent of bacteria cell wall is alcohol, waxes, pigment, and vitamin. Other constituent bacteria cell wall include alcohol हो सकता है, waxes हो सकते हैं, pigment and vitamin होता है. So next, this is overall about chemical composition of bacteria. Just you have to take a screenshot. And dry weight is about 50%, and 50% is it contains 50% of carbon, and 8 to 5% of 15% of nitrogen, and 2 to 15% of ash. So when we will calculate this it will be 70%, and 30% will be unknown. I don't know here because in our in our KK Bhalchandran book that's enough information is here. 30% maybe other than water is present there, I think personally. So this is about chemical composition of bacteria. Then thank you for watching this video. If you have any queries or like that, go in comment box and type. And if you don't if you want next part of this video, go in playlist, check out there. I had already uploaded the uh, fifth part of this lecture. So thank you, stay tuned with our channel. Thanks.